I'd like to thank the organizers for the invitation to participate in this event on gradients and brain organization, and the audience for your attention. My name is John Murray. I'm an assistant professor at Yale University with appointments in psychiatry, neuroscience, and physics. And I direct a lab which focuses on computational neuroscience. And so I'd like to describe two studies today from my lab. One on developing large-scale circuit models of large-scale brain dynamics, such as resting state functional connectivity, and the applications of those to study the effects of pharmacology on large-scale brain dynamics. With the specific test bed here, looking at a data set of resting state fMRI under LSD administration. And the second study will instead be on statistical modeling of large-scale brain gradients in maps, and how we can apply those to statistical analyses of gradients in neuroimaging. In particular, applications to brain map comparisons and gene set enrichment analysis. Okay, so a large motivation for my group is trying to bridge levels of analysis. And so, for example, when studying neuropsychiatric disorders, we're especially interested in symptom variation at the level of behavior and how that may be influenced by molecular alteration and therapeutics, which happens obviously at the level of receptors, which is a huge gap in variation. And so we can try to kind of piece in these levels of analysis to create mechanistic bridges. And so there's obviously huge literature developing neural biomarkers using functional neuroimaging to establish what are the patterns of neural features, be they maps and networks, which relate to symptom variation. Still a large gap to the level of molecules. And so we can kind of bridge that from the other direction. And my group and others are looking at mapping of gene expression and transcriptomics to look at the brain-wide distributions of different genes implicated in different pharmacological and disease processes to try to derive neural maps, which can be related to neuroimaging. We can bridge this gap even further by doing pharmacological neuroimaging with causal perturbations of the molecular systems and observing the impact on large-scale brain networks. And then we can make those mechanistic links more computationally rigorous by simulating dynamics in large-scale biophysical modeling and simulating the effect of pharmacology on given receptor systems and simulating the effects on large-scale brain dynamics, which in turn we can relate to neuroimaging. So the way this would look like is we're going to take this large-scale model derived and constrained to resting state fMRI, for instance, and we're going to simulate at some level of resolution excitatory inhibitory neuron interactions within a local microcircuit. And we're embedding that circuit within a large-scale network. And then to simulate the effect of pharmacology or molecular perturbation, we can use gene expression mapping, such as from the Allen Human Brain Atlas, to derive what the topography of the receptor densities are using the gene expression pattern as a proxy. To simulate differential strength of perturbation by a systemic pharmacological manipulation across the brain. And then we can also be informed by slice physiological studies to gain insight into what are the cell types and receptor sites implicated in particular synaptic or receptor alterations. And so we can then use this framework to simulate differential predictions for neuroimaging, be it fMRI or MEG, and test and constrain the model hypotheses. And so the test bed that we're going to be using here is a data set collected by our collaborators in Zurich on resting state fMRI with LSD administration. And our measure is going to be what we call global brain connectivity, which is merely the mean functional connectivity of an area. And what we see when we compare LSD to placebo is a very interesting topography across cortex of the change in GBC, in particular this large increase in visual cortex, which you see here. This is a very unique data set because what they found is that they were able to completely block both the neural and the behavioral effects of LSD by pre-administration of catanserin, which is a more selective antagonist of the serotonergic 5-HT2A receptor. And so we can see that here when we compare LSD to LSD plus catanserin, we get something very similar to the placebo condition. And so this gives us a very strong hypothesis for bridging from the systems level of neuroimaging to the molecular level. And that's that this neuroimaging map from pharmacological imaging should align with the gene expression topography for the gene HTR2A, which codes for the 5-HT2A receptor. And so when we compared this pharmacological imaging map to the gene expression maps derived from the Allen human brain atlas, what we found in line with our a priori hypothesis was that the HTR2A map had the strongest positive correlation out of a set of implicated genes, serotonergic and dopaminergic. And we can again compare that to the distribution across the full range of genes in the Allen human brain atlas for comparison. And so what we'd like to do on the modeling side is try to directly simulate the effect of the drug in a biophysically based model and see whether we can recapitulate the neuroimaging effects observed at the systems level. And so we have this model with excitatory inhibitory neurons embedded on each node. The interactions between the nodes are constrained by a structural connectivity matrix derived from diffusion-weighted imaging. We simulate synaptic and hemodynamic time series to create a simulated bold functional connectivity measure. And then we can simulate the effect of LSD through the 5-HT2A gene and the HTR2A, 5-HT2A receptor by relying on physiological studies which implicate neuronal gain and the transfer from input current to output firing rate being a neuronal site of modulation by this receptor. And so we can do this differentially onto excitatory and inhibitory neurons and have the strength of the gain change follow the topography of the HTR2A gene derived from the Allen human brain atlas. And therefore the strength of modulation follows that topography. Finally, we can then get a simulated functional neuroimaging measure, in this case, the delta GBC, and compare and fit that to the empirical measures. And so we have two free parameters here in the model, which are the strength of gain modulation onto excitatory neurons and onto inhibitory neurons. And we can then look within this 2D parameter space and see how that impacts the fit of the model delta GBC to the empirical. And what we found is that the model gives the best fit to the empirical when there is a net positive fractional change in excitatory inhibitory ratio, indicating a larger increase in gain preferentially onto excitatory neurons relative to inhibitory. And that is consistent with some more detailed physiological studies of the effect of the 5-HT2A receptor. So that's one conclusion consistent with the prior literature. And now we can look at what these topographies are. We have the empirical delta GBC, the best fit simulated model delta GBC, and we can get a nice fit to this cortical topography. And one really interesting thing is comparing this back to the gene expression map alone. And what we found is that whereas the gene expression map alone to the empirical map had a fit similarity around 0.4 something, here we're able to get up above 0.7 by actually simulating the neural measures which are being fit to in the dynamical circuit model. And so this suggests a framework of how we can bridge all the way from the molecular to the systems level by simulating neural dynamics and simulating the topographic patterns of receptors and their differential impact on different brain regions, and ultimately how that affects the brain patterns, in this case at the level of gradients. We can compare that to randomized maps, and it's somewhat unfair to compare our HTR2A map to a randomly shuffled topography, which doesn't look like the smooth gradients that we observe in the gene expression maps. And so we developed another framework for developing spatially autocorrelation preserving surrogate maps, which I'll describe shortly. And these have randomized topographies, but preserve spatial autocorrelation. And then we can refit the model with these different surrogate maps and compare our original HTR2A map to assess significance. Similarly, we can also compare our HTR2A gene to other implicated genes, serotonergic and dopaminergic, And here we found that the HTR2A map not only has the best fit, but also the most robustness of the fit within parameter space. Okay, and so to round this out with the second part of my talk, I'd like to talk now not about biophysical modeling, but about statistical modeling. In particular here, generative modeling of brain maps with spatial autocorrelation, which is preserved or matched to the target map of interest. And so here we may have empirical maps such as T1, T2, or thickness. And if we simply shuffle the values of this map, we get essentially white noise, which is an implausible kind of null hypothesis for testing questions about specific topographies of these brain maps. But this is essentially what the implicit hypothesis is when one does a standard conventional, for example, a Pearson p-value that doesn't take into account the autocorrelation structure. And therefore, we felt that there was a need in the field to instead use these spatial autocorrelation preserving surrogate maps, which can be matched in their spatial autocorrelation structure to the target map, but have randomized topographies and random patterns that emerge, and then use these to generate null distributions for our statistics of interest. And we can, again, kind of fit the spatial autocorrelation structure to the empirical map. And so to give one example of this, if here we're testing whether the T1, T2 map is significantly aligned with the thickness map, our assay-independent surrogate maps or a conventional assay-naive Pearson p-value would give very implausible p-values. But instead, if we use the assay-preserving surrogate maps, we can generate a plausible null distribution against which to compare our target. And for example, here, if we take two random assay-preserving surrogate maps and say, what is the fraction of pairs for which a standard Pearson p-value would give you a significant result, we find here that 93% of randomized topographies nonetheless would give you a significant p-value, but in this case being spuriously driven by the null hypothesis that the maps are completely independent white noise and lack spatial autocorrelation. And so this shows that spatial autocorrelation can substantially bias and drive spurious results in these types of map-map correlation analyses. So I don't have time to go into it here in terms of the algorithm of how we generate these spatial autocorrelation preserving surrogate maps. But you can read about it here in the paper that's in press. And we've released a software package called Brain Smash for generating these maps, which use basically a target map values and essentially a distance matrix between all the elements. And so this framework is very flexible. It can give you maps with full coverage and can be applied to both surface and volumetric neuroimaging data, dense as well as parcellated, and cortical as well as subcortical. And so we think that this may be a valuable tool for the community. And as one final application of how this could be used, I'd like to talk about gene set enrichment analyses. And so these have become increasingly popular in studying large-scale gradients in neuroimaging. And so the kind of standard approach is to take your map of interest, compare it to many gene maps, for example, by looking at the alignment or correlation, selecting the most correlated genes, and then doing a gene set enrichment analysis on different ontology terms to see whether those are significantly enriched for these particular genes relative to a random set of genes. And the problem here is that this does not actually test the null hypothesis of whether these enrichments are being driven by the specific cortical topography of the map of interest or neural topography, and not being spuriously driven by the fact that these maps are smooth and autocorrelated, which in itself might preferentially select some genes over others and drive these enrichments. And so what we propose is an assay-corrected gene set enrichment analysis, which has two steps. The first is deriving assay-preserving surrogates from the target map, and then constructing a set of p-values across the genes for that set of assay-preserving surrogates. And so if we look at that, for example, what we can see is that in this case of spatially autocorrelated surrogates, we actually get significant enrichment in a number of different gene set ontology categories, which are not being driven by any particular topography, but are merely enriched for genes which have spatial autocorrelation themselves. And so you can see the problem here, which is that if one did this on a single map of interest and observed enrichment in these categories, you would not know whether this is due to the specific topography of the map itself or merely due to the fact that it's spatially autocorrelated. And therefore, the second step is to then transform the p-value for the empirical map into an assay-corrected p-value by comparing the observed p-value against the distribution derived from the assay-corrected surrogates, assay-preserving surrogates. And that gives us an assay-corrected p-value. And in this case, for the T1, T2 map, what we find is that many of the enriched categories, according to the traditional analysis, can be explained merely by the fact of spatial autocorrelation, because they are also enriched equally for the assay preserving surrogates. But we can also identify which categories we get a significant corrected p-value for, indicating that this enrichment is preferential for our empirical target map, in this case T1, T2, even relative to assay preserving surrogates. And so we think that this may be a way to prevent the kind of spurious enrichment which may be observed in these gene set enrichment analyses. Okay, so to wrap up, I've described two studies, one of circuit level modeling, where we've tried to bridge from the molecular to systems level, bridging from transcriptomics to circuit network dynamics to pharmacology. Ultimately, we would like to bring that to the level of behavior. And in the second part, describing statistical analyses of gradients and how that can be, number one, spuriously driven by the spatial autocorrelation, which is such a prominent feature of large-scale gradients, and how those can be addressed and corrected through generative modeling of autocorrelation matched surrogates. And finally, that we've released an open access software platform called BrainSmash to facilitate these analyses. Finally, I'd like to thank my collaborators at Yale, in particular the lab of Alan Antichevich and the University of Zurich, and really highlight this is primarily the work of Joshua Burt, a senior PhD student in my lab, as well as thank my funding sources. Okay, thank you for your attention.